Biochemistry. Last minute revision points. Acetoacetate, beta hydroxybutyrate, and acetone are not substrates for gluconeogenesis. Major contribution towards gluconeogenesis is by alanine participating in glucose-alanine cycle. It is also known as Cahill cycle. UDP glucose is the key substrate for glycogen synthesis. Beside liver, gluconeogenesis occurs in kidney, primarily in cortex. NADPH plus H plus is required for fatty acid synthesis in cytosol. The most common enzyme deficiency in man is glucose 6-phosphate dehydrogenase that leads to hemolytic anemia. NADP is used as a coenzyme by glucose 6-phosphate dehydrogenase. HMP shunt pathway produces CO2. Galactosemia commonly is majorly due to the deficiency of galactose 1-phosphate uridyl transferase. Galactosemia can also be due to the deficiency of galactokinase and uridine diphosphogalactose 4 epimerase. Pathognomic of galactosemia is oil drop cataract appreciated in slit lamp examination. Cataract in galactosemia is due to accumulation of galacitol or dulcitol. Treatment of galactosemia is lactose-free diet. An enzyme involved in the catabolism of fructose to pyruvate in the liver is glyceraldehyde 3-phosphate dehydrogenase. In hereditary fructose intolerance, there is a defect in aldolase B which is predominant in liver. In fructosuria, deficiency is fructokinase. Sorbitol pathway. Aldose sugar is converted to ketose sugar by isomerase. Production of fructose in seminal fluid is by following steps: glucose 6-phosphate to glucose sorbitol to fructose. The end product of one-stage fermentation is ethanol. After alcohol ingestion, acetaldehyde accumulates in liver that is not typical of glycolysis or the citric acid cycle. Goic test is used for hematuria. Untreated diabetes leads tissues to use fatty acids as main fuel. Ketoacidosis without glycosuria is seen in prolonged starvation. Retina is completely dependent on glucose as fuel. The problem of regenerating NAD plus from NADH for cytoplasmic process by using mitochondria is solved in the most energy efficient manner by intercellular shuttle systems. Malate to citrate shuttle, also known as citrate malate antipode or malate oxaloacetate shuttle. Glucose and galactose is a pair of epimer with respect to C4, while glucose and mannose are epimers with respect to C2. Routine examination of the urine of an asymptomatic pediatric patient showed a positive reaction with clinic test, a copper reduction method of reducing sugars, but a negative reaction to glucose oxidase test. A sucrose is least likely to be present, assuming a single elevated saccharide. Fructose is likely to be present. Essential fructosuria is asymptomatic. Mucopolysaccharidosis are inherited storage disorders. They are caused by defects in the degradation of proteoglycans, that is glycosaminoglycans. The most likely cause of hemolysis is a less than normal level of reduced form of glutathione. In a well-fed state, acetyl-CoA obtained from diet is least used in the synthesis of acetoacetate. In oxidative phosphorylation, the ATP production and respiratory chain are linked by chemoosmotic methods. Thymine is coenzyme for pyruvate dehydrogenase complex and alpha keto dehydrogenase complex. The synthesis of glucose from pyruvate by gluconeogenesis requires the participation of biotin. The conversion of pyruvate to acetyl-CoA and carbon dioxide involves the participation of lipoic acid. The reaction catalyzed by phosphofructokinase 1 is the rate-limiting reaction of the glycolytic pathway. Substrate level phosphorylation seen in reaction catalyzed in succinate thiokinase. In fasting, increased levels of alanine indicates increased breakdown of muscle protein. Major determinant factor in gluconeogenesis in liver is ADP. Fluoroacetate inhibits the combination of acetyl-CoA with oxaloacetate. Basically inhibits citrate to cis aconitate conversion step. After overnight fasting, level of glucose transporters are decreases in adipocytes and muscles. A patient present with hypoglycemia and hepatomegaly with renal enlargement. Most likely diagnosis von Gilger's disease. Thiamine requirement by body depends on consumption of carbohydrates. Phosphorylase B is inactivated by glucose. Mucopolysaccharide hyaluronic acid is present in vitreous humor. The function of 2,4 DNP is uncoupler of electron transport chain. Hypoxia increases glycolysis due to 2,3 DPG, which causes activation of pyruvate dehydrogenase complex. Phosphorylase B in the muscle is normally kept in inhibited state by AMP. Glycogen in muscle cannot contribute directly to blood glucose. First substrate of Krebs cycle is acetyl-CoA. Enzymes of glycolysis are present in cytoplasm. Glucose finally metabolized to carbon dioxide and water in mitochondria. Inorganic phosphate is used in enzymes of glycolysis, phosphoglycerate kinase. Final common pathway of metabolism of carbohydrates, lipids, and protein metabolism is TCA cycle. Cori-Forbes disease is due to the defect in debranching enzyme. Glucose is transported in pancreas through which GLUT2 rapid transporters. Epimer of glucose is C2 mannose and at C4 galactose. Cyanide affect respiratory chain by non-competitive irreversible inhibition. Hyaluronic acid is the major proteoglycan of synovial fluid. GLUT2 receptors insulin independent. The energy of glycogenesis is provided by UTP. Leg disease is due to pyruvate dehydrogenase phosphatase deficiency, where accumulation of glycogen is seen as the dephosphorylated form of PDH complex is only active, and failure of activation due to the phosphatase deficiency leads to improper breakdown of glucose. It is due to oxidative phosphorylation defect, where NDUFS gene mutation leads to defect in complex 1 of ETC. Transport of glucose into adipose cells is stimulated by insulin. Retina completely depends upon glucose as a fuel source. Postprandial utilization of glucose is by enzyme glucokinase.